Right. This thing that I've drawn here is the basic, and I mean very, very, very basic the very, very basic structure of von Willebrand factor. So you can think of it just as a big, long protein that's going through all these different domains like this. Okay. And I've labelled up the important subunits that we need to take into consideration. So I'm saying the important subunits because it's impossible to draw it all out. It's huge, this thing. It's 20,050 over that amino acids long. It's a big old protein, okay? But the point is that each of these different domains has got specific functions. I'm just going to go through the really important ones that you need to remember. Okay. I suppose the follow-up question to that should be, what do they bind to? Well, they're either going to bind to, where shall I draw this? Again, you think about it. We'll think about our vessel. Okay, this time I'm going to leave the top of the vessel and I'm just going to draw the bottom of it like this. There's trauma there. Okay, so we can get rid of that, and we can replace that with some collagen. Okay? The von Willebrand factor is either going to be binding, choose my colours carefully here, is either going to be binding to that collagen or to the thing that we're going to talk about next, which is our platelets. Okay? They're going to be binding to, these domains are going to be binding to specific things on this. So for the things that are covering a platelet, which I'll introduce you to later, called glycoproteins, I'm going to draw like little hands like this. Okay? You see them? There they are. Okay? I'm going to draw little hands like that. So if you see me drawing these are platelet glycoproteins, and they're the thing that's going to be kind of binding onto the different domains of von Willebrand factor, or the von Willebrand factor is going to be binding directly to the collagen that we've exposed when we carried out the vascular trauma. Okay? So, starting from this end over here, okay, we've got, this is the CK domain, C2, C1, then we've got a chunk of domains together which are just labelled B1 to 3. Then we've got slightly larger units then called A3 and A2. There's a little bit of space in between, and then we come to A1. D3 and D-tick, D2 and D1. I don't know why that's not D4 and D3, but that's just the way that it's called. So, let's start with A1. Okay? Now, A1 is going to bind to a particular platelet glycoprotein. Okay, now the particular glycoprotein that it binds to is called glycoprotein, okay, G1B, okay? That's what that is going to bind to. So A1 is going to bind to platelet glycoprotein 1B. This A3 domain here, that is what's going to go ahead and bind to the collagen. Later on, okay, so you can notice that what we're trying to do is we're going to try and take this von Willebrand factor and anchor it down to this area where it needs to do its work. Later on, this subunit here, C1, is going to bind to a different platelet glycoprotein. Okay? Now the glycoprotein that it's going to bind to is called glycoprotein, okay? Glycoprotein, G. I keep wanting to write another letter because we used to call these GP, but now we just call them G. Glycoprotein, you can see how I keep wanting to write the second letter. Glycoprotein 2B, 3A. Okay, so it's going to bind to that one. But, as I already said, there's going to be some order to the way that this happens. Okay? So, I suppose the next question is, well, where does all of this come from? Where does this von Willebrand factor thing come from? And the answer is that it's formed within the Golgi. Okay, so it's formed within the Golgi, like most things are. Okay, and then what you get are these things which are called pre-pro von Willebrand factors, and they kind of bind together. Okay, and they form something called multimers, which we'll come to in a little bit. These things, okay, are then kind of transported off so that they can be stored elsewhere. And they are stored deep down there in the subendothelial layer. And they're stored in something, and I want to make sure I get this right. So I'm going to make sure that I've actually looked at my spelling, and we can draw them in. So we'll draw them in like this. Okay, they're called, and again, I'm going to try and, where possible, do this in capital letters so that you can actually read my handwriting, because I have appalling handwriting. They're called Weibel-Palade bodies. Okay, Weibel-Palade bodies, and they are what are going to be responsible for storing von Willebrand factor down in those sort of, sometimes you find them actually in the endothelial cells themselves, and sometimes they can be just below that in those subendothelial layers. Okay, now that's not the only place that they get stored. So inside these platelets, okay, there are storage granules, and in the alpha granules, you also find von Willebrand factor stored there, okay, in the alpha granules of the platelets. They can, however, only release that von Willebrand factor when the platelets themselves have become activated. So there are steps that things need to go through before we can start releasing this stuff. Okay? Suffice to say, von Willebrand factor is an incredibly important protein for this whole process. In terms of stimuli as to what can bring about von Willebrand factor's release, there's a couple of basic things that you can think about, and obviously one of them that we've just talked about is trauma. Okay, so from the endothelium itself, okay, trauma, so we'll write this up here, trauma. So von Willebrand factor is then expressed from endothelium, etc. But something else happens as well. From the Weibel-Palade bodies, okay, these guys can release von Willebrand factor in response to various different stimuli, okay, one of which is physical exercise, and another one is adrenergic stimulation. So anything that is going to get your adrenaline going is going to start releasing von Willebrand factor, and it's going to do that regardless of whether you've got any trauma in the system at all. So it's constitutively expressed from the endothelium, okay, and that happens when there's trauma or anything like that. However, adrenergic stimulation, and I suppose you could say that also goes hand in hand with physical exercise, is going to get the von Willebrand factor released from the Weber-Palade bodies as well. When platelets become active, they release it from their alpha granules. Now, the problem that I suppose we can think of is, and I've kind of touched on this already, is that this von Willebrand factor is a bit of a big beast, okay? So if I create myself some space here, then I suppose I can try and illustrate it slightly better. So I want you to think of von Willebrand factor as being kind of like a wacky waving inflatable arm flailing tube man. So you know the guy from Family Guy, you know, Al Harrington. You know, hi, I'm Al Harrington of Al Harrington's wacky waving inflatable arm flailing tube man emporium. I am currently overstocked on wacky waving inflatable arm flailing tube men. This is posing a huge fire hazard. So each of these wacky waving inflatable arm flailing tube men have got to be sold, and I am going to be passing this saving on to you. That guy. Practice that one. So when the von Willebrand factor gets out, okay, and it starts to bind into that collagen, at this point, pause the video, and without looking back on anything, see if you remember which of the domains that it binds to the collagen with. Okay? Are you back? Good. Okay. So the von Willebrand factor is going to be binding into that collagen, and what it's going to do is going to kind of unfold with the direction of the blood flow. So you can see that, you know, it's at this point in time. Let's get rid of the platelet altogether. Let's get rid of the platelet altogether so that we can illustrate the blood flow thing. So here we go. So we got the blood flow coming through like this. Okay, and the von Willebrand factor, big beast though he is, okay, it's going to be kind of going like that. And it's going to be waving around like a wacky waving inflatable arm flailing tube man. It's big. And what we don't want, because the way I usually describe platelets is they're not very bright. They kind of just go kind of rolling along the bottom of the endothelium like that. What we don't want is a platelet ending up clotting up there. So this von Willebrand factor needs to be kind of cut down to size, okay? And it is cut down to size by something called, where shall I put this? Let's get rid of the blood flow. Okay, it's cut down to size by something called Adams TS13. This is a very, very long acronym, and I don't expect you to remember it, so just call it Adams TS13. Now, Adams TS13 is basically Pac-Man, and what it's going to do is it's going to come along and it's going to cleave von Willebrand factor at that A2 domain. Okay? So it's going to cleave it off there, and it's going to cut it down to size. So these fragments, okay, these fragments split off. They go into the blood flow, into the middle of the vessel where the flow is quickest, and they get carted off to the liver, crunched up, and sorted out. Okay? Now, What this also does is that Adams TS13 now, is that it basically catches any von Willebrand factor that is excessively large, because one of the problems that we have with von Willebrand factor is that it likes to bind to itself, and it makes something, and this can happen sometimes in the rough endoplasmic reticulum, called ultra large von Willebrand factor multimers. So Adams TS13's job is basically to come along and chop up anything that is going to be too big and cause a clot that is going to be too large within the vessel. So we're keeping everything right down to the size, or in other words, as we call it in the game, we localize. Okay, so that's what I want for slice 4 for you to know about von Willebrand factor. Okay, there's quite a decent amount of information just on this one board, but it's the key stuff that I want you to kind of have your head around. Now, I've made a big thing about informing you which domains are responsible for doing what. So I really do want you to pay attention to that, because it's important. The next thing that we're going to talk about is platelets, and what they do and how they work. So we'll do that in the next slice. In the meantime, happy hunting.